Thank you. So our next speaker is Christabel Chan. So Christabel is working on her PhD at the National University of Singapore. She's interested in the mechanisms responsible for the selectivity of three-finger toxins and snake venoms. So today she'll be talking about alpha and omega using neurotoxins to investigate nicotinic receptors. Please go ahead, Christabel.
Hi, everyone. Okay. Yeah, so for those in the UK, good morning, and those everywhere else, good afternoon. So welcome to my presentation, Alpha and Omega, using neurotoxins to investigate nicotinic receptors. So yeah, I'm Christabel. I'm a PhD student currently at the Protein Science Lab in the National University of Singapore. So let me just, yeah. So today I'll be talking about snake venom and why are snake venom toxins so interesting, right? Because snake venom is actually a cocktail of bioactive compounds containing anticoagulants, presynaptic neurotoxins such as phospholipase A2s, as well as postsynaptic neurotoxins and vasodilators like natriuretic peptides. So in nature, snakes use snake venom as a tool to capture prey and for feeding, but with the advance of technology, we have been able to utilize these bioactive compounds to create medicines and drugs for commercial and pharmaceutical purposes. So one type of toxins from snake venom is known as the three-finger toxins, and these three-finger toxins make up a large proportion of some elapid snake species venom, such as the coral snakes. So this pie chart represents the different types of bioactive compounds, and as you can see here in yellow, the three-finger toxins make up a large part of it. So more about the three-finger toxin family. They are a family of non-enzymatic proteins of 60 to 74 amino acid residues, and they have a conserved structure of 3 beta-stranded loops extending from a small globular hydrophobic core, like 3 fingers extending from a palm of the hand, hence the name. And this hydrophobic core is cross-linked by 4 conserved disulfide bridges. And most of these three-finger toxins have potent toxic effects. So if you look at the diagram here, these show different members of this three-finger toxin family, and you can see all of them have this three-finger shape. And their toxicity depends on their targets. So some of them target the nicotinic acetylcholine receptor, some of them the muscarinic receptors, L-type calcium channels, so on and so forth. So they have conserved structure, but a variety of targets. So now I want to talk about a subclass of the three-finger toxins known as the alpha neurotoxins. So these toxins are postsynaptic, so they target the receptors on the postsynaptic membrane, and they are the principal neurotoxic components of the elapid snake venoms. The alpha neurotoxins can come in short chain or long chain form, and both have high affinity to the muscle type nicotinic acetylcholine receptor found at the neuromuscular junction. However, only the long-chain alpha neurotoxins have a high affinity for neuronal nicotinic acetylcholine receptors. So why are these alpha neurotoxins so important? Because they have been used as tools to study the nicotinic acetylcholine receptors. So in 1970, Shonju and his colleagues observed that alpha bungarotoxin from Bunger's multi-cinctus, which is the many-banded crate, bound irreversibly to the then uncharacterized cholinergic receptor. So this led to the purification and eventual characterization of these nicotinic acetylcholine receptors. And in the decades that have followed, toxins have been continued to be used to study the structure-function relationship of these nicotinic acetylcholine receptors. So more about these receptors. They are from the cis-loop ligand-gated ion channel superfamily, and they are made out of 5 polypeptide subunits assembled like the staves of a barrel. And you can see here in this diagram, they are made out of extracellular domain, transmembrane region, and intracellular domain. And the agonist binding site, the acetylcholine binding site, is located at the extracellular domain in the interface between the subunits. So between for example, the alpha and the gamma subunit over here, but I'll elaborate on that later. And they are permeated by ions such as sodium and calcium and so on. So because multiple different types of subunits can be combined to form one receptor, multiple subunits combine in different ways to produce nicotinic acetylcholine receptor diversity. So on the left over here, you have the muscle type peripheral receptors, which are located at the neuromuscular junction. So these are made up of alpha, beta, delta, gamma, or epsilon subunits. Then over here, on the right, we have the neuronal receptors, which are located in the central nervous system. So these can be homomeric, so only the alpha subunits, or heteromeric, which contains both alpha or beta subunits. So like I mentioned before, the agonist binding site is located at the interface between the alpha and non-alpha subunit. So for example, the alpha 4 and the beta 2 subunit over here. And this is a close-up crystal structure of this interface where the alpha unit provides the principal binding phase. So you have loops A, B, and C contributing to this binding site, as well as loops D and F contributed by the complementary phase, the non-alpha subunit, contributing to the selectivity in this binding site. And then this is the cartoon diagram showing the important residues, mostly aromatic residues that are involved in the acetylcholine binding site. So because of this diversity, different subunits can combine to produce full receptors that have different physiological and pharmacological properties. So some of them have a higher or lower agonist affinity. And studies have shown that various subunits are also involved in different neurological diseases, such as alpha 7 and alpha 4 beta 2 in epilepsy, Alzheimer's disease, schizophrenia, so on and so forth. So this leads to the question, right? So how do we identify the distribution of each subunit and their physiological function in the human central nervous system? And how could we also maybe treat neurological diseases in a specific manner? Well, we suggest that one solution to this is to make probes specific for different nicotinic acetylcholine receptor subunit interfaces. So now I will introduce the toxin of interest, which is short-chain alpha neurotoxin, Aerobutoxin A. So this toxin comes from the Laticoda semifasciata, which is the black-banded secrete. This Aerobutoxin has a high affinity to the torpedo marmorata nicotinic acetylcholine receptor. So this is the electric ray. And this particular receptor from the electric organ of the electric ray is homologous to our human fetal muscle receptor type. So in 1999, Antil and their colleagues wanted to study the various conserved residues in the short-chain alpha neurotoxin family. So they mutated these conserved residues to find out which are responsible for the high affinity for the receptor. And they found that certain, they observed that certain mutations actually led to a high affinity binding interaction in the picomolar range and a low affinity binding interaction in the nanomolar range. So they have they observed that there were 2 KD, so 2 dissociation constants, whereas the wild type, the native Aerobutoxin, only had a single dissociation constant. However, the authors did not investigate the reason for the different binding sites, and they only reported it as an incidental observation. So from their incidental observation, this led to our hypothesis, which is that these particular Aerobutoxin mutants may be able to discriminate between the nicotinic acetylcholine receptor subunit interfaces. So one high affinity binding site and one low affinity binding site. So now I'll just jump straight into the results and kind of like the findings that we had, and also a bit of what kind of experiments that we did. So in order to find out the effect of Aerobutoxin and their mutants on the nicotinic acetylcholine receptor, we use electrophysiology, where we inject human cRNA into the Xenopus laevis oocyte, so it's the frog, in order to allow the oocytes to express the human nicotinic acetylcholine receptor. So in this experiment, we inject 2.5 nanograms of human cRNA in each oocyte, and after 48 hours, the human nicotinic acetylcholine receptors would be expressed on the surface membrane of the oocytes. So then we would conduct the electrophysiological experiments using the 2 electrode voltage clamp setup, where there'll be 2 electrodes that will be clamping the membrane voltage at a particular level, so negative 80 millivolts, and then we would have 1 electrode to inject current, and then another electrode to measure the internal potential. So when the ion channels open in response to the agonist, this would cause membrane depolarization. So the amount of current needed to be injected to maintain the membrane at negative 80 millivolts would be recorded as the current, and it should directly correlate back to the amount of membrane depolarization. So this is the machine that we're using. So for these experiments, we used Aerobutoxin that has been expressed using recombinant expression using bacteria in order to obtain these toxins. So first, for the fetal muscle type expressed in the oocytes, these are the traces for the acetylcholine-induced currents. So on the left, we have the reference. So just acetylcholine that has been introduced, and then the following membrane depolarization. So in color, on the right, we have the antagonist inhibited currents, and we can see that the acetylcholine-induced currents are inhibited in a dose-dependent manner for the Aerobutoxin wild type. So for one of the mutants, where arginine 33 has been mutated into glutamic acid, we can see again that there is also a dose-dependent inhibition based on the dose response curves. So again, on the left in black, it's the control, and then in color, it is with exposure incubation with the mutant. So because arginine 33 is one of the well-conserved functional residues, when you mutate it, you can see that the IC50 goes down, which means that more toxin is needed to inhibit the receptor, and this is to be expected when you mutate a functionally conserved residue. So for the adult muscle type, again, on the left in black, it is the control current with just acetylcholine, and then in color, it is with the exposure to the toxin antagonist. So again, there is dose-dependent inhibition, and with the mutant, you can see that at the start there is some dose-dependent inhibition, but once we hit 40 nanomolar of the mutant, there is a plateauing of the dose response curve. So what could this mean? So for this particular mutant, we call it R33E mutant. We recall that the fetal dose response curve is quite standard. It's a single sigmoidal. However, for the adult receptor, there is this plateauing of the dose response curve. So this could indicate that instead of a normal sigmoidal dose response curve, it could actually be a double sigmoidal profile that we are observing over here. So where one curve is for a higher affinity interaction and one curve is for a lower affinity interaction. So if you look between the fetal and adult, the higher affinity interaction profile kind of matches that of the fetal. And between the fetal and adult receptor, the common interface is the alpha-delta interface. So this could mean that the higher affinity interaction is due to, it corresponds to the alpha-delta interface, and then the lower affinity interaction, it could belong to the alpha-epsilon interface. However, more data needs to be collected in order to substantiate this observation. So now I'll move on to the second part of my talk, which is about omega neurotoxins, which is a novel class of nicotinic acetylcholine receptor antagonists. So I'm going to be talking about two of them today. So we have OH91 from the king cobra, characterized as an omega neurotoxin in this paper published in 2015, and exactin from Hemectus hemectus, published in this paper from 2016. So if you look at the sequence alignment over here, omega neurotoxins actually lack the conserved residues of alpha neurotoxins, but still have neurotoxicity. So using Aerobutoxin A as an example of the short chain alpha neurotoxin, you can see that OH91 and exactin, they lack the conserved residues such as the serine, glutamine, and lysine over here. And if you look at this graph over here, so on the y-axis, it represents the normalized response of the acetylcholine-induced currents of these particular nicotinic acetylcholine receptors that have been expressed in oocytes as well, same as the earlier diagrams that I've shown. So when OH91 was applied, we can see that the muscle types, so over here, both adult and fetal from the rat, they were almost completely inhibited, both by 100 micromolar as well as 30 micromolar when this toxin antagonist was applied, as well as the rat neuronal alpha-3 beta-2 receptor type. And also, OH91 also weakly inhibited the human neuronal alpha-3 beta-2 and alpha-3 beta-4 receptor types. And the dose response curve shows that OH91 inhibits the receptors in a dose-dependent manner. Similarly, exactin also inhibits the nicotinic acetylcholine receptors, hence it has neurotoxicity, however, in a slightly different manner from OH91. So this graph over here also represents the relative current amplitude or the normalized response of the acetylcholine induced currents from the receptors expressed on the oocyte. So you have OH91 in black and exactin in red. And you can see that from the bar graph over here, the OH91, even when OH91 was applied, the level is almost 1, right? So there's not much inhibition for human alpha-7 and human alpha-9/10 neuronal receptors from OH91. However, exactin almost completely inhibits these two neuronal receptors. So this shows that exactin has a distinct selectivity towards the neuronal human alpha-7 and human alpha-9/10 nicotinic acetylcholine receptors, unlike OH91. So what leads to this selectivity of the neuronal nicotinic acetylcholine receptors? So looking at the sequence alignment over here, both of them share almost the same residue. Now the residues or primary structure in the loop 2. However, the loop 1 and loop 3 have very different residues. So this leads to our hypothesis that the unique loops 1 and 3 of exactin may confer this unique neuronal receptor selectivity. Hence, 2 chimeras were prepared to test the nicotinic acetylcholine receptor subtype selectivity. So chimera 1, which has loop 1 of exactin replacing loop 1 of OH9, as well as chimera 2, which has loop 3 of exactin replacing OH9. So what we will do moving forward is to test if loop 1 or loop 3 of exactin or both of them combined confers selectivity to the human neuronal alpha-7 and alpha-9/10 receptors. And then following that, we would do site-directed mutagenesis on the chimeras to determine which are the specific residues influencing subtype selectivity. And once we have an understanding, we can further study the fundamental mechanism behind how the toxins have subtype selectivity for the different kinds of receptors, as well as develop probes in order to target specifically different types of receptors so that we can gain a greater understanding of neurological diseases and maybe develop them into pharmaceuticals. So with that, I would like to close my presentation, and I would like to thank Prof. Kini Manjunatha, who's my supervisor, as well as my whole lab for their support. So thank you.
Thank you, Chris. If there are any questions now, you can post them in the chat. Okay, so actually I do find one here from Dr. Michelle, yeah. Oh, you see it too, Chris? Or should
Yep, I see it, yeah.
Okay, so as a double sigmoidal curve observed in the adult nicotinic receptor, could this be due to differential binding sites on the receptor?
Yes, so that is what I think is happening, because so the double sigmoidal would mean that there is one higher affinity interaction and one lower affinity. So we are hypothesizing that this is actually what the Antil et al. Actually observed in their paper in 1999, which is why they reported two different dissociation constants. So actually, this is quite exciting because this kind of puts us closer to investigating the hypothesis, whether they can actually differentiate between the two different subunits within one single receptor. So yes. Yeah.
Nice. Okay. So we have another question from Dr. Rohit Patel. Have you looked at how long it takes for the receptor to show recovery after omega neurotoxin binding?
Yeah, so in the paper that was published, the paper that I talked about was actually published from our lab. They actually show that the binding is slightly reversible, so it can be washed out, but it takes about 3 washes to, I think, get back about maybe 20% of activity in the acetylcholine-induced currents for the receptors. Yeah, so it's slightly reversible. Yeah.
Okay. I've got another question. On one of the graphs, I think it was with the channel mutants with Aerobutoxin.
Yeah.
When I think the lowest concentration tested was 0.1. Did you go lower than that, and did you see any increase? So it plateaus at about 75%, 70%.
Yeah.
If you went lower, did you see even more recovery, or was that the actual plateau?
So I haven't been able to repeat this experiment, but so if I went lower, I think I probably could, get. Are you talking about the left side of the dose response, or the, like increasing?
In the left side of the dose response. It's
right.
With the adult type ones on these 2 slides here. So
right. So this one, right?
The top one, sorry. So it's about 60% the plateau on the 39.3 nanomolar.
Yeah. Oh, you mean the top one? Yeah. Okay. Yeah, I think I would definitely like to repeat this experiment because I think the first point wasn't kind of well collected, so it doesn't reach to 100%. But I mean, there could be a plateau here as well, but I'm thinking not very likely. Yeah, but yeah, I do.
Do you normally see a lower response when you use the adult, because the fetal muscles are quite large current, and this is
yeah. So it's because they are expressing different subunits. So when we were expressing the adult one, the currents were a lot smaller because of some technical error. That's why the noise to signal ratio is a lot higher in the adult type compared to the fetal type. Yeah, but again, this is my preliminary data, and I still have a lot of optimization to go. Yeah, so yeah. Thanks for the question. Yeah.
All right. I have a quick question for you, Christabel. So the toxins, the Threefinger toxins, are you producing them from recombinant expression, and what system do you use to do this?
Yes, so I'm using E. coli to produce all of the toxins, both the alpha neurotoxins as well as the omega neurotoxins. So we use the shuffle cells, and the vectors that we use are like PET22B vectors with the T7 promoter. Sorry, not T7. So we just use the shuffle cells, and then to obtain the folded proteins, we use buffer oxidation. So once we purify the proteins, we reduce them and then we refold them again in a controlled environment. And then we do circular dichroism to check whether they are folded correctly with the proper beta strands. Yeah.
Okay, thank you.
Maybe a question from me. Christabel, hi,
Yep,
Christabel.
yep.
I
Hi.
just want to find out like what kind of control condition that you include in your protein folding. Is it you use some kind of glutathione? Or
Yeah.
is there
So
is-
as in control, as in when we do the circular dichroism, is it or?
no, what I mean is what kind of oxidation method that you
Oh.
use in the during the
Oxidation.
folding? Yeah.
Yeah, so our redox reagents, we use oxidized and reduced glutathione in methods that have been published by people in like the 1990s that worked on these similar three-finger toxins. So I personally use a phosphate buffer. And then with varying amounts of ratios of oxidized and reduced glutathione. Yeah.
Okay. Right. Thanks for
Thank
that.
you. Thank you.
All right. I think we have come